Growing a human heart from a single cell may seem like science fiction, but scientists at the Gladstone Institute at the University of California, San Francisco have taken a huge step forward by producing the first three-dimensional beating human heart chamber. Previously, it had been possible to produce a two-dimensional sheet of beating heart cells, but to really gain an understanding of heart formation in a developing fetus, and perhaps most importantly, how drugs given to women during pregnancy may affect this development, a three-dimensional structure was needed. By treating stem cells with drugs and then confining them to a very small spherical geometry, Bruce Conklin and his team have managed to grow their very own three-dimensional model of a human heart.
The cells around the edge became fibroblasts, a particular type of cell that you use to heal wounds, and then only in the very center were cardiac cells that beat. What this is forming is more of a little organoid, is what we call it, where there's beating cells, but there's also multiple other cell types, and that's what makes it so interesting, is that these cell types are somehow talking to each other and somehow collaborating in some way so that they can actually make this structure that we didn't expect.
So it's almost like they're trying to form a heart.
That certainly is the impression. They're heart cells, they're forming a cavity, so it could be a model of how parts of human development occurs, but it's certainly not a real human heart in the sense that there's probably many things that we're missing. We just have a simplified version with just one chamber, but having it in a controlled way where it happens the same way over and over again, we can start asking questions about how do these cells talk to each other. So once you have a system which is reproducible, then you can do experiments to say, to break it in some way or to enhance it in other ways.
What are the applications of this work then?
The most obvious application of the work is to study human development. How do cells actually, form a heart is something of basic interest. And also, the most common form of birth defects is actually cardiac defects. But the other application is that we can expose these developing human microchambers to drugs which are thought to cause developmental defects, specifically of the heart. And in fact, one of the key experiments in this study was to use the drug thalidomide, which is notorious for causing birth defects. When we exposed these cells to the thalidomide, they had a dramatic change in the morphology so that you could see that it was altering the developmental process in this microchamber. Thalidomide was tested in rodents before it was tried in people, and there were no cardiac defects in the rodents. I think that more and more we're thinking, how do we get tests which use real human cells so that we can actually make safer drugs? And in this case, if, say, you turn back the hands of time and you had this sort of test, perhaps you would have discovered that thalidomide was dangerous before it had gone on to be given to people.